Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stoliarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings and welcome to our Virtual Enlightenment Salon of April 10th, 2022. We are pleased to have a fascinating and unique conversation in store for you on cat longevity. Joining us is a distinguished panel of U.S. Transhumanist Party officers and members, including our Director of Visual Art, Art Ramon Garcia, our Legislative Director, Jason Geringer, our Director of Longevity Outreach, Ben Ballweg, and our Technology Advisor and Foreign Ambassador in Spain, Dr. Jose Cordero. And our special guest today is Alexandru Voda, who is a final year Doctor of Philosophy student at the University of Oxford in the United Kingdom, though today he joins us from Romania. He is a transhumanist, a longevity advocate who has worked on longevity science for a decade and a half, and he is a published researcher. Today, he will speak to us about a project that he currently is engaged in regarding cat longevity diagnostics. So welcome, Alexandru. It's a pleasure to have you join us today. What aspects of cat longevity are you studying, and what can you tell us about this fascinating subject?
Thank you for having me first, and this is really a pleasure to talk to all of you. I think there are many reasons why cat longevity is an interesting topic to look into, and why diagnostics are a subtopic of that are a nice pick. So firstly, there are many reasons why you could go into pet longevity projects. One of them is that you care about your pets. Obviously, I have a cat. I wish that they'd have a longer lifespan, not just myself. But there's also a lot of other reasons, right? So if you look at most of the startups that are going into longevity, if they're choosing the vet space and are sort of asked to justify about it to investors, one of the reasons the investors may hear is regulatory burden. So one of the things that's pretty difficult with human trials is getting through the approvals by FDA to run the initial trials, ongoing regulatory filings even after the drug is approved, and so on and so forth. Whereas the veterinary landscape is a lot lighter. And there may be pros and cons to that. So obviously one cons to that is that because it's less regulated, it's a bit more risky, meaning when you enroll your pet in a trial, you can be slightly less confident about how much work has been put into assuring that that drug is safe and sound for the prescription that it's being trialed for. But also it has a lot of cons, which is a lot of pros, which is having less regulation means that you can quicken the innovation rate, and because the burden that you have restricts not only the budget of the company, restricts also a bunch of the iterative things that you need in terms of trial drugs out. So with all of that boring stuff out of the way, in terms of why you'd pick cats, there's also some funny reasons why you'd pick cats in terms of longevity research. It's because they're longer lived, they chase lasers, and Also, there's very few companies pursuing anything related to non-dog stuff. So if you look at the longevity vet space, you'll see Rejuvenate Bio by George Church and people from his lab. You will see Selina Haliwa's Cellular Longevity Incorporated company. I think it's Loyal for Dogs. And all of these companies have huge funding, and it's really really great to see that. But at the same time, it sort of begs the question of why we're avoiding half the pet healthcare market. And that's the sort of gap that we're trying to fill. And the interesting thing about diagnostics is there are all of these aging clock methods out there, which I'm sure some of you might be familiar with. And I hope you're not annoyed at me for patronizing some introductory discussion on it. But essentially, aging clocks are a combination of markers, levels of proteins or RNAs or DNA methylation and metabolites, anything that can tell you something about the status of the healthcare in some organism. And they're widely used and accelerating in use as well, mainly because of several reasons, including the fact that survival trials are very expensive. So following up treated versus non-treated groups takes a lot of years and takes a lot of funding, whereas it's slightly easier to look at biomarkers that are correlated to something that you're interested in, like longevity or health span or the status in the disease that's heavily linked to mortality, like heart failure and so on. So the interesting thing about clocks is there's a huge fight at all times on all topics related to clocks. For example, whether they should be used in trials or not. If you just browse Twitter for a while, you will see at least 3 fights every afternoon between very hand people in the fields, and I think that's good. That means that there's a lot of innovation going on. That means there's a lot of back and forth on how to do certain things, and maybe there are multiple ways that you could do certain things. And we certainly think that we can do better clocks in terms of chronological aging estimation, and in terms of mortality estimation, and so on and so forth in cats. So as a first plugin, I co-founded a company called the Cat Health Data Science Corporation. It's currently incorporated in Delaware with Alexandru Voda, my awesome co-founder. And we've got a few other people joining us very soon, and we're looking out for investors. We are currently in talks with VitaDAO and a bunch of other investor panels. And yeah, I think that's a pretty long introduction to it, but for the moment, I think that's it. And what I can tell you about the cat longevity diagnostics. Oh, some other additional thing is our venture is supposed to be split in 2. So we're going to have various projects that we're going to pursue in an open source, collaborative, free access way, patentless, and usable by any people on the internet that can follow the specs of the algos and laboratory protocols that we're developing. And then there's also going to be a commercial side. So on the non-commercial side, we just got funded through Gitcoin by a lot of generous donors of crypto for about $10K worth of $10,000 worth of sequencing and other sort of costs for a trial to improve aging clocks in cats. And obviously, depending on what a panel thinks about this, we can dive into details on the specifics of the clocks, or on the company and the commercial side, on longevity in general, and politics, and yeah.
Excellent. Well, thank you, Alexandru, for those introductory remarks. And I've linked here to the page on Gitcoin where essentially you have a description of your project. Would you say this is the most comprehensive source for people to visit if they want to learn more, or is there another page somewhere where you have even more details or an overview of your plans that you would be able to share?
This is definitely the most in-depth resource for the open source projects of the company. We are gonna increase the detail in there as we go, and we're gonna update that link as we're gonna participate in further rounds of Gitcoin. But in terms of the commercial side, the only help I can provide is telling you that we're doing more than the open source projects. So most of the slides that we have are investor oriented and have proprietary stuff on them. But I guess I can give you a very short summary on that, maybe like 30 seconds of it. So essentially on the commercial side, we're interested in further developing the clocks, but also looking into therapies, so things that can modify the clocks and can modify the mortality, and also have a few other measurements outside of the clocks in terms of whether they work or not for particular prescriptions. So for example, one prescription that happens in a lot of cats is sarcopenia. 38% roughly by the latest research is the rate at which elderly cats suffer of sarcopenia. Mild or severe sarcopenia. So keep in mind, it's not all of the old cats have severe sarcopenia, as in they can't move around, but they have mild muscle loss, which can be, we hope, mitigated by certain therapeutic approaches. And the sort of main therapeutic approach we want to go on the commercial side, and this is a differentiator between us and other longevity companies in the pet healthcare area, is that we're not seeking to find new aging drugs. So obviously there's a lot of small molecules out there that modify lifespan and health span, from rapamycin to metformin and so on and so forth. The question we're trying to answer on the commercial side of the startup is whether specific combinations of known off-patent drugs act in synergy. So if they have a positive interaction, say you have a drug that has that improves lifespan by 3% and a drug that improves lifespan by 5%. The question here is whether together they improve lifespan not by 5%, not by 3%, but whether they improve the lifespan by 20% together, just because they have a positive interaction. So we were trying to find whether there's any high effect size, so anything between 30, 50, 70% improvement in various healthcare measurements in elderly cats. And we want to do that mainly because cocktails of unpatented drugs are patentable. And that way we can also get some revenue to sort of fund the other open source projects that we want to pursue.
And on the open source side, what will your focus be? What do you anticipate publishing, and how can people collaborate with you on that side if they wish?
So on that side, I think the most use, firstly, if anyone has any idea on helping us, please feel free to email me. My email is alexandru.voda at thecathealth.com. So I'm just going to type that in the private in the comments soon. And in terms of specific help that we could use, obviously, outside of funding, which we will never reject, is expertise or access to Oxford nanopore technology. So one thing that we want to use more in the future is long read sequencing technology, which is not currently used by most of the clocks on the market. For example, the Steve Horvath clock is based, most of them are based on arrays, which is kind of like a plate with tiny pieces of DNA or RNA or so on and so forth attached to them, and imaging on that. We want to use sequencing because we can find new molecules, so we don't have to fix them on a plate and decide beforehand what's important or not. And there's a couple of advantages in that technology and we want to exploit that other people haven't. But obviously, we're not experts on all of the aspects of nanopore technology. So anybody that's specialized in there is welcome to email me, of course. Also, we're very interested in collaborating with as many veterinary doctors as possible. So right now we're in talks with about 20 veterinary doctors across the UK and Romania. And we're looking to expand in the US, and we actually haven't talked to any veterinarians in the US as of yet. And the reason why that's useful is because obviously the blood collection and all of these initial procedures are done by veterinary doctors in cabinets, and we need collaborators on that side. And also, it's nice to have a clinical input in the sort of drug lists that we're currently filtering down to having run with the trials later on. So yeah, any help in that, or any help in anything else, if you have an idea of how we can get more funding, or if you have an idea related to how we can reach European funds, public research council funds, and any of that, we're very open to collaborators on that. So yeah.
Yes, thank you very much, Alexandru. And if anybody listening to this salon would like to engage in those kinds of collaborations, I have posted Alexandru's email here. And now for a bit more of a lighthearted interlude, here's a joke from Mike Lazine. Why are cats cooler than dogs? Answer: you never hear anyone saying that a person is a cool dog. No, they say cool cat. So I am biased. I am a cat owner myself. I have 2 cats who are approaching the age of 12, and I clearly want to maximize their longevity. And if I had 2 dogs approaching the age of 12, they would be in trouble already, because one of the advantages that cats have over dogs is that they do live a bit longer. So I'm curious, as a scientist, how would you characterize the differences in longevity between cats and dogs? Now, intuitively, average lifespans for cats are longer. How much longer? And what is the variation in cat lifespans as compared to the variation in dog lifespans that has been observed?
So it is very hard to characterize that, mainly because both in dogs and in cats, there's a lot of variation between breeds of cats. That's the first thing that comes to mind. Even in dogs, if you look at pugs versus Great Danes, they have very different diseases that they're being afflicted of. Most dogs have heart failure as a mortality cause, whereas pugs specifically have more leaning into respiratory diseases. And so you can see, depending on how evolution shaped their bodies and metabolisms, you get very different outcomes, average outcomes. In cats, one interesting factoid is that actually a lot of cats die in car accidents and traumatic events like falling from a tree or stuff like that. You might have heard, and seen with your own eyes that cats always land on four paws. But actually, they expose themselves to way more mechanical risk than dogs tend to do. So that's a very tragic thing, and it happens to a lot of people. A friend of mine just lost his cat about several days ago. So also they fit more places than dogs, which is again another reason why they have mechanical risk on lifespan. But overall, I think the major causes of disease, as far as medical statistics journal, veterinary statistics journals go, are CKD. Chronic kidney disease is a very big thing in cats, and it happens a lot with age. And then all of the other things that you see in humans and in dogs as well. So like heart failure, cancer, and a bunch more like neurodegenerative diseases and so on and so forth. I think there are big factors in lifespan, including like how wealthy the owner is. That makes a very big difference in the environment that the cat is brought in. It makes a whole difference in their diet and lifestyle and activity and access to veterinary care. Obviously, that's one of the issues that the startups is trying to estimate when we're trying to reach investors and sort of give them a realistic scenario about what the potential revenues could be, is how much people can spend on their cats. Because obviously most people prioritize their own healthcare over their cats. Healthcare is not super affordable, even if you live in in a state healthcare sort of area. And I think the average spending on healthcare in cats is about $200 a year in the United States, or something less, $150, something around that area. Whereas in other countries, it's significantly less. It gets closer to $50. So that makes a huge difference in how best you can treat fractures that they suffered during trauma, which a lot of cats go through. I think probably like 20% of the cats or 15% of the cats suffer of mechanical trauma, like injuries, strained muscles, broken ribs, fractured legs, and so on. I think it'd be best to cite a paper on this. I have a favorite one, which I keep citing. It has a really nice table. So I'm going to mute myself just for a minute to seek it, and then I'm going to post it.
Mm-hmm. Yes, thank you. And while Alexandru searches, I will point out a few resources that we have shared. First of all, the oldest cat ever recorded, and there's a Wikipedia entry on her, is Cream Puff. Cream Puff lived to 38 years and 3 days. She lived from August 3rd, 1967 to August 6th, 2005. Now, this is remarkable in that this is a much longer lifespan than most cats reach. Generally, when cats die and we learn about it, they're in their late teens or early 20s, at least anecdotally. But this Cream Puff was able to live to essentially twice as long as many cats. And the question that I have is this a plausible lifespan for cats to reach if they received the same kind of medical care that humans get? If a human becomes ill and suffers some sort of chronic condition too that might be expensive to maintain, generally there's no argument, at least in the Western world, that money should be spent on this, whether it's a private system or a governmental system. So if cats had a similar standard of care, do you think many of them would live into their 30s?
I think most not. So most of the cats, even with very good care, would not. It's still determined by a bunch of very stochastic, very random processes. It also depends on an individual's genetics, right? Like even in humans, where people have this programmed aging versus non-programmed aging fight at all times on Twitter and in papers and everywhere. It's easy to forget that things are sometimes in the middle. So, for example, human longevity is somewhat heritable, and it's kind of the same for cats as well. Like even in cats, you don't expect that all of the genotypes will live extreme lifespans, even if they have all of the favorable environment possible without new interventions. Obviously, if you take enzyme replacement therapy and gene therapy and new small molecule drugs and localized delivery of specific therapies and significant improvements in the technology of current medicine, I'd say yes. But without significant improvements, I wouldn't say that the mean lifespan would change a lot. And it's easy to forget that human lifespan didn't significantly extend without radical changes in medical technology, from vaccination to hygiene to improvements in cancer care and therapeutics in general and drugs. So I would say no. Also, I would say that it's very hard to take these one-off cases. So it's very hard to say this is the oldest recorded cat, and I'm sure that she's I lived with her in my house for like 38 years. I can't say for certain that happened because I didn't live in the house. I don't have any indication about the sort of rumors about Marie-Jeanne Calment. If you remember, there's a lot of fights about whether she actually lived that long, whether it was inheritance fraud that her daughter committed, that sort of stuff. And so I'm trying to avoid it, even though, like all of you, I care mostly about maximal lifespan, because I think that as long as I put all my resources in lifespan medical lifespan enhancing medical technology, that I can fare better than the average lifespan provided in general. But obviously the goal would be to extend medical technologies to where you don't care about one example, to where you don't have to look at Marie-Jeanne Calment or the longest lived cat or the longest lived dog, where you can just say, oh, by pure chance, I think as long as I have these therapies on hand, I can think about myself living way longer than just 70, 80, 90 years old. And it's really hard. There's a really nice paper. I'm sorry. Did you manage to post the one I put in the private?
So it's currently being shown here. So that's the paper by Dan O'Neill and others from 2015.
Awesome. So that's like a completion on what we talked before as well, where it shows that trauma is like the leading cause of mortality in this sample of several thousand cats. Like 118,000 cats. And then it's followed by CKD, renal disorders, and other things like that. And then cancer, heart disease, etc. So that's a really nice paper. Another paper I'd mention is one I'm going to put in the private chat very soon. I think most of the longevists know about it. It's a paper where they've shown that villages where there's evidence of extraordinary longevity in humans, tend to, not all of them, but some tend to be correlated with increases in inheritance tax fraud, which is an unfortunate thing to have. And so the question is whether we should trust extreme longevity or median longevity. And obviously, I want extreme longevity to be something that we can touch, something that we can experience in our lives. But it's probably best to improve the median one with improved technology, improved drugs, improved therapies, and so on. And then think about the extreme longevity in just individuals as opposed to groups. Maybe there will be some libertarian yelling at me. Just know I'm on the same boat, but I'm just spitting some 2 cents on that.
Yes, well, thank you for sharing your thoughts on this. Now, I will note with regard to extreme human longevity, now Kane Tanaka in Japan has reached the status of the second oldest human ever, the second oldest verified human. She is 119 years and 98 days old, and she just became the second oldest human ever today. The third oldest human was Sarah Knaus, who died on December 30th, 1999. She was 119 years and 97 days. So even if Jean Calment's history might not be authentic, and I don't know if it is or isn't, we have someone who is likely perhaps to get to 120. So there is this outer limit right now, given the capabilities of medical technology, that is around 120 years for humans. And I do think it would be an immense victory for our movement if that limit could be reliably breached, even by several people. If more individuals are verified to exceed Jean Calment's record, that will raise attention to the promises of longevity science and convince people that death by a certain age is not inevitable. So I do think there's value in seeking to extend maximum lifespans. But what I'm curious about is in terms of the comparison to cats. So Cream Puff died at 38. Is 38 then the analog of the human 120, in the sense that the reason why most cats die much earlier than that is the reason why, say, in the Middle Ages, most humans died much earlier than the 70s or the 80s that they reliably reach today? Or is Cream Puff's age more similar to the human 200, where cats are capable of being much more extreme longevity outliers as contrasted with humans, where if humans do get good healthcare, good sanitation, according to the current standards, if they are able to avoid wars, pandemics, etc., then even these days, without too much effort in terms of lifestyle, most people can live reliably into their 70s. So with regard to cats, are they in the equivalent of the human Middle Ages, or are they in the equivalent of the human 20th century in terms of their baseline conditions that allow them to live to a certain age fairly reliably?
We hope that they're in the Middle Ages right now, and not in the sense that we hope they stay there, but in the sense that we think that because there's lower regulatory burden, and because there's more opportunities to innovate in that space, we think that they might be on the verge of significant life extension of the kind that we've seen in centuries ago in humans. I think humans will follow as well, but you have to think about how people allocate risk, and most of the people don't want to allocate risk on themselves, except very famous examples like Elizabeth Parrish, like Nobel Prize winning gastroenterologist who proved, I think, a type of bacteria that could grow. There was a type of bacteria that could grow in gastric juice. And there's a bunch of other people that are an exception to that rule, but nonetheless, that's a rule. And I think that's a rule for anybody who isn't a biohacker. And most of the people aren't biohackers. And so I think there's a lot more innovation to be had in a very short time span in cats right now. Otherwise, I do my best to try and do stuff in humans. Although I know there's a lot of human work that's amazing right now. So there's a lot of work in a bunch of therapeutic categories, from blood factors, a lot of people looking at individual markers that they can upregulate in blood, to people that into cell therapies, into gene therapies, into a bunch of these things that they're slowly carrying in humans over, I guess, over decades, since books have been written, from End Engaging to a bunch of other very famous books. You will see that innovation in humans has been kind of slow. And a part of the reason for that is that risk allocation is very different in humans. We tend to take way less of a risk with people, with our own kind, and with essentially people that can live very long, right? Whereas with cats, when you get a 16-year-old cat that has sarcopenia and CKD, the worst case scenario is she dies a few months earlier. And so risk allocation in people's minds is very different. And I think that's going to be the driver for why dogs and cats are going to have a lot of medical innovation. And the really interesting thing there is, that saying that if you're a mouse, we have very good news for you. One of the reasons why a lot of mouse research is failing is because the sample sizes for experiments are very small, and the mice are very uniform, and they live in kind of like the same conditions. That's for good reasons, because you want to have reproducibility. You want to see in the same conditions, the same genotype, you get the same effect. But the question for drugs in humans and a lot more complex animals is, how can you find drugs that have large effects, no matter the environmental conditions or no matter the genotype? And I think you're going to see that a lot in cats and dogs, mainly because the risk allocation is much higher, and because the treatments are ultimately carried in non-uniform animals. They're coming from a wide variety of backgrounds. And so you're going to only see and only focus on the effects that actually are reproducible, no matter the conditions. And also, I'm going to post this paper on inheritance fraud versus extreme longevity.
Yes. Yeah. And I will share that link shortly. I'm curious if you think that it would be feasible, along the lines that you are studying, to develop treatments that could be disseminated at least to a significant portion of cat owners, such that, say, by 2030, a lot of house cats could live to age 25, for instance, fairly reliably.
So obviously we want to scale up as fast as possible. I don't think we can make promises as to how long the cats will live, because obviously some therapies work better for some things, right? Like if you're going to develop some novel methods of treating CKD, age-related chronic kidney disorders, then that's obviously not going to extend the lifespans of cats that don't have that type of disease. And I think that's the ticking bomb with aging, which is like a lot of people are trying to say, "Oh, I'm just going to extend lifespan with this drug, or I'm going to do that with that therapy." And really, I think even when you look at very general treatments like rapamycin, metformin, and so on, really you're targeting an agiotype. I can't remember what paper it was that coined this term, but it's a really nice term that I think more people should use. Agiotype means a subtype of aging in terms of what diseases you get, what sort of metabolism wreckings are going around, or what sort of, if you're a programmed aging sort of believer, which you may or may not be, I don't know, what sort of sub part of the clock is getting its wind back. So I think we can't promise, cats are going to live 30 or more by 2030. And I don't want to do that sort of operating rate prediction because it's really hard to hold without data. And one of the things that I care a lot about is showing people some work related to longevity that they can reproduce and that they can try on their own 100 cat sample size trial, and they're going to get the same result. And I don't think I can right now say that there's any therapy that's going to take for sure cats to 30 years old or so. But if we see anything that extends lifespan even by 40 or 30%, then we're going to pursue that. And obviously, anything that's lower than that, we're not interested because it's really widely available in terms of general, like more multi-agiotype therapies, kind of like aging retardants. But I would certainly hope that we can extend the lifespan of cats by a lot. And if we see, like, if we have reliable data in our trials of combinations of known aging modulators to see if they have positive interactions, where you have a 3% effect, but 5% effect not being equal to 8% or just 5% or something like that, but equal to 30% or more, I would certainly hope that at least some of the cats from thousands of cats being treated with that therapy from there on, I would certainly hope that some of them will live very extreme lifespans. I would hope that they reach 40 and that have an amazing, never before seen thing. But the thing about that is you cannot predict it. You just have to work on it. Kind of like how if you asked Professor Cynthia Kenyon whether she would have found 2 mutations in DAF2 and DAF16 or whatever other genes back then that would double the lifespan of C. elegans, then before that finding, she'd be like, I don't know. I really don't want to promise anything. Or maybe she would be all like, I don't think that that's possible. But it's the thing that we can say is that there are reproducible life extensions by specific small molecule drugs, specific enzyme replacement therapies, and so on and so forth. And then the optimistic scenario is combinations of those in various doses are going to increase lifespan by a lot. The medium scenario is like they're going to extend lifespan by some amount, and then like the worst case scenario is actually they work on the same thing at all times, and there's no lifespan extension. In which case, everyone who's been all like, "Oh, I told you cryonics was the lead," or mind mapping and transcendence is really the goal for longevity, would be very happy about this, but I'd be very sad about it because that'd be my favorite scenario where you can essentially extend lifespan by a lot. So I think, that's about it.
Yes, and absolutely, that would be my favorite scenario as well. I would rather not have this biological existence be interrupted because I do have various concerns, as I've expressed in prior virtual enlightenment salons, about whether one's I-ness, that which makes one the same individual, would be perpetuated with these other techniques, whereas with biological life extension, it clearly would be. Now, I did post the link to the paper that you mentioned by Saul Justin Newman entitled Supercentenarians and the Oldest Old Are Concentrated into Regions with No Birth Certificates and Short Lifespans. And I just wanted to get your thoughts on this, Alexandru, before we move on to the rest of the panel. And that is with regard to common claims made about the blue zones, for instance, that people there get plenty of fresh air and outdoor activity, and they have robust social networks, and they have good dietary habits. Do you think that's a significant contributor to the observed longevity in those areas to the extent that it is observed? Or do you think a lot of the observed longevity could be explained more by these kinds of statistical artifacts or correlations with other activity we don't necessarily want to see, like the potential to forge a birth certificate?
I think it's a mix. I think there's both inheritance fraud and certain things about certain villages and certain areas, diets, lifestyles, and so on, that modify lifespan by a lot. The hard thing there is to distinguish how much in between them, because it's clear that it's both of them. You can have a model that has one at 30% and the other is 70%, and then another model is completely different. And while I'm very interested in statistics and I study statistics at my PhD a lot in terms of figuring out how to weigh things mathematically, I'm not a specialist in that area. The only thing I know from people that work in the field is that there is controversy around how much each of them weigh. And I don't think anyone says there's, or maybe if they do, they're very silly about this, that there's no benefit in specific areas of Japan, for example. Japan has a lot of villages that have extreme longevity groups. And I think also Ashkenazi Jews have specific villages populated mainly with Ashkenazi Jews have extreme longevity as well. There's a bunch of other groups that are very interesting to study, but there's one challenge that a lot of statisticians will tell you about, which is observational studies have issues with causality tracing. So you don't know whether specific factors are causal to that, or whether when you have a genotype or an environment that makes you live longer, then you also do some other environmental thing. So you don't know, for example, a very nice example is let's say Marie-Jeanne Calment lived very long. I don't want to fight the people that say that she didn't. I don't want to fight the people that say that she did. Whichever way it goes, let's assume that she lived 127 or something like that. Then some people might go on and say, well, she smoked a lot and ate a lot of chocolate. So just throw the diabetes and the lung disease sort of issues away. They're not an impediment to longevity. And really the answer there is, okay, so you looked at an observational thing where maybe you had this observation of the data. You cannot assert causality. You cannot know that there are other things that affected the outcome or that the direction of the effect is right. That's why you want to have interventional studies. And again, it's hard to have interventional studies in humans. It's not hard to have interventional studies in vets, mainly because of the politics and the way people structure their lives and their governments and their regulations and so on. And so in terms of in terms of saying stuff like there's things in these villages that prolong their lifespans, I'm certainly interested in hearing about the data, but I'm also very wary about focusing only on that as opposed to doing the most important thing, which is interventional trials. And I think, if there's a piece of advice in there, take more natural teas, do more exercise, go to church, hang around with people, have a community around, that sort of stuff. If that's some advice that comes out of the paper, fine. But if we're starting to say, oh, that's really the only meaningful thing that we can do about longevity, or that's the thing that we should focus on, or we should fund X more studies on these observational village studies, then my personal answer would be maybe you should stop doing that, because there's much more value taking comparisons of randomized interventional double-blind stratified designs where you're asking villagers for a long time to do whatever treatment. Maybe you look at survival, or maybe you look at aging clocks, maybe you look at some biomarkers, maybe you do something else, but anything you do there is easier to assess in terms of causality than these sort of observational cohort studies. So I think they're nice, but for some people their value is overstated, and obviously there are some people for which their value is understated. They say, "Oh, these are worthless," and I don't think that they're worthless. But it's just about the focus. So I can remember my mom talking a lot about these. People eat this specific berry, and I was like, "Oh, come on!" Like maybe, but there's no, like, the study doesn't say that for sure this is what happens. This is an observational study. You have to give that berry to a randomized group and placebo to a whatever other group, and then you can say, this is the true effect, this is the direction, and it's et cetera and et cetera. So yeah, I think that's my final thought on extreme longevity in villages and studies of that.
Yes, thank you. And our friend Didier Cornell points out in the chat that Jean Calment lived to 122 years. I've linked here to the Wikipedia page. Her lifespan was 122 years, 164 days. So what's fascinating is that Kane Tanaka, if she lives for another 3.5 years, would be able to exceed that lifespan. So we shall have to wait and hope. And if she does exceed that lifespan, then perhaps Jean Calment's story will look more plausible to the skeptics as well. But now let us go to Jason Geringer. Jason, I know you have a number of questions, so please feel free to proceed with whichever one you think is most fitting.
Yeah. Okay. The kind of cats. I heard that there's a breed that's the most common breed, maybe even, is from like northern Egypt or northern Africa and stuff. Is that the kind you use? Well, first of all, is that true that that's like the most common breed? Is there, and is that the kind you use, or does it matter? Yeah.
I think it depends also by country. So different countries have different preferences in terms of breeds that they buy. There are countries actually that don't have any preference. They just have a lot of stray cats. I think that's the case for Romania. And I think I don't know. Do I remember right that the Egyptian cats are the ones that have very little fur, right? Like they're very wrinkly and
no,
yes, the Sphinx cats, they're called.
I think that's a different breed. The northern Egypt ones are not. They're like, I think the ones that the wild ones now look a lot like that breed, and so like my cat looks a lot like that breed. They're the kind that got I don't know, the stripy stuff. But I was wondering how exactly the experiments go. I mean, which cats you use. I mean, is there a way for somebody to get involved in an experiment with their own cat, even if it's the control group or something? I'm wondering how that open source stuff that you was talking about works.
So right now, for the open source trial, we're going to go very local and very small sample size because we need to do some power calculations and so on to figure out how many more cats we're going to need with the technology that we're going to assess. So we haven't received the funding yet to start the trial. We're going to receive it on the 15th of April, like all of the other longevity grants on Gitcoin. Just a little bit of self plug right here. We were third place in the longevity category, which was really amazing. And once we receive that funding, we're going to go very local. We're going to assess probably around 20 to 40 cats around Bucharest and around Oxford in the UK. But as soon as the small scale trials are over, which I expect should be in about 2 or 3 months, at least the first phases of the project, then we're going to extend. And we're still debating about how exactly to extend. So depending on how much funding we have and depending on how the team agrees to do the things, we're going to have trials where the consumers fund the trials. So all of the tests are paid by the consumers, and then they receive all of the results that we get, and we do analyses at the global scale where we have access to all of the data, whereas you only have access to your cat's data. The alternative is if we get enough funding, we pay for the trial, even for people in the States and so on and so forth. So if you want to come in the trial, you can email me. And as long as we have enough funding in about 1 or 2 months from now, when the first initial testing phase is done, then we may be able to fund the test for your cat.
Yeah, I think that's a viable thing. I mean, if someone was to say, "Hey, be part of this experiment, we'll give you these, the drugs for your cat or whatever," but then they would have to keep accurate data. I mean, how are you going to get just the general public keep accurate data? And speaking of that, you were saying that the fact that the cats jump around and they're cats, they get hurt because they kill stuff, and that is a high percentage, and I'm wondering, is that out of, you just mentioned the cats around Romania, wouldn't, if we're just talking house cats, like my cat that never goes outside, wouldn't that be way different, and wouldn't maybe we want the data to represent the cats that are probably going to be getting your funding eventually, which is probably, regular owners that, keep their cats inside, and in which case the data from trauma of stray cats wouldn't be valuable to that group. You know what I mean?
Yeah. I know what you mean. So there's 2 questions there. Let me unpack it a bit. So in terms of cats inside versus outside trauma, the differences in sort of disease prevalence in trauma versus chronic kidney disorder and sarcopenia and whatever other things elderly cats suffer from. Obviously, we're going to focus on household cats because then we have a point of contact, which is the owner. We did debate at some point trying to go for shelters, but unfortunately, a lot of shelters in Romania don't have very good treatment ethics for cats, and we don't want to get associated with that because we don't endorse it. A lot of cats are mistreated in shelters, and mainly because of poverty. Like, there are many excuses if you want to make them, but the principal reason why we'd go for household cats is also what you said, which is differences in the sort of diseases that and sort of physiology and health that status that they have. And so we only aim to work through vets. That's in terms of the administration of the drugs. Obviously, we're open to collaborate with vets across the world. So if you have a veterinary doctor for your cat that you're familiar with and you feel like you can communicate easily with them, we're going to have a few employees soon, as soon as we get funding, to sort of communicate with veterinary doctors or people that want to participate in the trial across the world. The second issue that we have there is shipping. So because the project is two ways. The first one is the open source trying to improve the diagnostic side. And then the second side is the therapy trial side. So in the therapy trial side, it's going to be the difficulties are regulatory approvals for those drugs in those states. Not all drugs are approved everywhere, even if they were FDA approved. So we have that one issue. And on the diagnostic side, which is a separate, so just to give you a timeline, the diagnostic side is going to happen in the next month to 3 months at most.
And that's the part that outside open source can help with. And so the open source isn't going to be on the okay, I get what you mean. What does the diagnostic side entail? I'm sorry to interrupt you. Go ahead.
So on the no problem. So on the diagnostic side, the open source side is we're trying to improve markers of overall health in cats. So obviously there's aging clocks. There's a Horvath mammalian clock. It's pretty expensive. It's more than $100 per sample, especially if you only send one sample. That's very expensive. We tried to get quotes for about 20 samples at a time, and they were still very expensive. So what we're trying to do is both bring the cost down while maintaining some accuracy on the blood clock. And then we're trying to get a few other measurements, such as how deeply the cat sleeps, like REM versus non-REM, the activity levels of the cat. So it turns out if you put a bracelet, a collar on a cat, you can get a lot more measurements than how heavy it is or the waistline or stuff like that. And the aim that we're putting out with the open source project is combining all of these different health measurements into one. And most of the aging clocks really only take into account at most some extra blood markers, like if you have high albumin or something like that. And so we want to take into account not just the classical BMI, sex, etc. things. We want to take into account activity levels, patterns of sleep, a bunch of other markers that we can do machine learning on and sort of develop better, more accurate clocks. And then
those collars would be accurate. You wouldn't have to be depending on the owner to give you accurate information. You could get
exactly. And the blood clock is the same, as in we don't depend on the laboratory prowess of the owners. But on the treatment side, which is going to be the commercial thing, obviously we still need collaborators and help and so on. So you can email us if you want to participate in the trials. And as I said, depending on funding, we might be able to do a thing where we pay the therapies for the cats, and that's probably the biggest aim for the diagnostics. It's still in debate, but for the therapies, we're definitely trying to subsidize everything, but it's still going to have to go through a vet because, as you said, it's highly difficult to figure out if the owner had time on whatever evening, whenever the drugs had to be administered. So you need to get a very reliable measurement of that. And I'm not saying the vets are definitely more reliable than pet owners, because a lot of pet owners are very reliable, and there are some unreliable vets out there. But it's just an average rate that we are trying to improve there, and that's why we're going to have to go with vets. So if you want to collaborate with us in the therapy trials, which I think are going to start in a few months from now, they're not going to start now on the 5th or on the 15th of April. And if you want to participate in that in a few months, email me now or then and we can discuss, finding a vet in your area that you're willing to collaborate
I'm curious if you know Kent Kamish, or if you've seen our salon with Kent Kamish. I heard you mention something about needing more tech or something, about nanopore, and he has this console that he's been working on about nanopore, and he does or was wanting to do some similar projects on mice. This could be possibly, maybe room for collaboration there. I'm just wondering if you knew or were aware of him and his stuff at all.
I might have heard. I think is it the Devil Box or something?
Demon Pour.
Yeah.
Demon Pour 64.
Okay. Yeah.
I think they've changed the game since then, but yeah.
Yeah, I've heard of these guys. They're on most longevity groups that I'm in. I think John Marlowe, who put me in contact with Kennedy. John is also working on a startup that's extending nanopore technology by automatizing specific steps and the pipelines required for the nanopores. We're going to work with him in the future as long as he and his brother has available time. We're up to getting in contact with anybody. So if you know them and you feel like they have enough time, because obviously people that run companies can be very busy, and without taking equity in hours, they may or may not be interested. But if you think that they can help us, definitely put us in contact with them.
I think that these guys could possibly help each other. Is what I'm thinking. Not only just like him help you, but I mean he's almost kind of doing the same thing in a way but on a smaller scale. And if you just do that same thing for cats, there's a lot of times it's a win-win situation. It's possible.
And for those who have not watched it yet, I've linked to our Virtual Enlightenment Salon with Kent Kamish, which we held on June 6th, 2021. Quite an extensive conversation, 2 hours and 48 minutes. So please check it out if you have not done so. We have some interesting feedback from our audience as well with regard to the diagnostic pet collars. Alan Crowley writes, collars would be like a Fitbit for the cats. And Daniel Twet says he could see pet Fitbits becoming a hot seller. So maybe people would be interested in purchasing diagnostic measuring devices for their cats. Now, we also had an interesting comment from Mike Lazine, who observes that domestic cats live far longer than jungle cats or cats like lions. And he wonders what causes that, especially because lions are bigger and most jungle cats are bigger as well. And generally, size positively correlates with longevity. So why do these relatively small house cats live longer? Is it just that they are in more favorable circumstances where they don't have to deal with very hostile environments at times, or is there more to it than that?
I think you summarize what I'm going to say very well in the last couple of words. So I think it's a lot in the stress side. Obviously, cats don't have to hunt buffaloes, and buffaloes, not buffalo. I'm making stuff up now, but it's a bit on the stress side. So it's an observed fact that animals that don't have natural predators tend to live longer. Because they don't have to reproduce as fast. They can. There's this germline soma, disposable soma theory of aging, where essentially, which is still discussed. It's evolutionary biologists disagree on it still, but the essential gist of it is that if you're not under evolutionary pressure from predators and so on and so forth, then you don't have to reproduce faster at younger ages. And so, for the species to survive, and so what happens is you expend more energy, more metabolic effort into properly structuring your soma. And I don't know if that's true or not. I'm just giving a very uneducated opinion on this, but I think it's mostly stress, and I think it's also access to healthcare. So wild animals will very often eat infected meat. Will eat very unhealthy things just to survive, whereas cats don't have to do that. And they have access to there's actually a lot of debate here as well, because people are saying sometimes on in research papers that maybe cats are better off if they eat raw meat or infected whatever, but or whether it's just the dry grains that are bad for their health or not. But right now, even if say it's healthy to eat some raw infected meat from some dying animal or something like that for a cat, say just by random, the infection goes mainstream, goes in their blood, they get sepsis, they are gonna die. And there's really very few lion doctors out there, which is what keeps their lifespan low. And again, trauma in household cats is way lower than trauma in any wild animal. And so remember from that paper, a lot of deaths in stray cats and in cats in general that get to a veterinary doctor are trauma related, not age related diseases. So like 12.2% is trauma, 12.1% is CKD and other, and then downstream from there, less and less other age-related diseases. So cumulatively, aging is definitely a huge cause in cats. And the reason for that is they have access to antibiotics. They don't have to hunt for a living. And also they don't have poachers, I think, at least not in my neighborhood. And that's about it.
Mm-hmm. Yes, thank you. And indeed, you articulated some of the reasons why I am a strong advocate of keeping cats indoors, and my two cats are purely indoor cats, so they do not get exposed to the trauma that they might encounter if they were to hunt outside. Besides, I live in an area where there are some predators such as coyotes or hawks that I see on quite a frequent basis, and cats, let's say, do not have high chances when they encounter those kinds of predators. And I cannot imagine that eating infected raw meat would be a net positive for longevity. I am generally in favor of the hypothesis that foods that are designed specifically with the health of an animal in mind would, at least on average, be more likely to contribute to their longevity or prevent their premature death than just whatever random piece of meat they might find in the wild. Now, we have a question from Jose Cordero, who wonders, are rapamycin treatments on your agenda for cat longevity trials?
Yes, they are. I think rapamycin is one of the high effect size drugs out there. I mean, high effect as it goes, right? Like nothing is high effect enough, really, because it doesn't provide. I think we all of us agree that rapamycin is not a universal panacea that really solves aging at all. But it's a high effect size drug, and it's reproducible. As far as meta-analyses and research papers go, and it's on the radar. The question is not just whether we select individual drugs, like say, we pick this one and we say this is my favorite drug, this is my favorite pathway. The really interesting question for cat therapies that we're pitching investors right now is what combination of them works best. And you might have seen some of these approaches across the last 1 or 2 years or 3 years. You might have seen Steve Horvath labs. It wasn't just Horvath lab. I think a recombinant growth hormone plus metformin that reversed aging clocks by 2 years or so. You might have seen an increased thymic volume, right? And the thymus is a big problem with aging. There's actually a lot of people at the institute I work in Oxford. Right now in Oxford, I don't work in this moment in my PhD on age-related diseases, but a lot of people are working on thymic involution there at the Kennedy Institute of Rheumatology. And you might have seen George Church's Rejuvenate Bio gene therapy, which targets Klotho and 2 other genes, I believe. And so you see companies pursuing combos of aging targets. But the thing that we're really interested in is scaling this up a lot. So we want to take a lot of treatment groups and a lot of control groups and compare real permutations of drugs that we suspect from previous computational analyses that they act in different pathways. So if you take a database like DrugAge made by the Joao Pedro Magalhães, I don't know if I'm pronouncing this right, from Liverpool. He's a very top biogerontology, bioinformatics researcher. That database has about 1,095 drugs that modify lifespan in various papers. And it's about 2,000 papers. And if you take all of those 1,000 drugs approximately, and you try to run them through a pathway analysis, gene ontology, disease ontology, so on, these sort of computational guesses at how the drugs manage to have an effect, you will see that a lot of them act through the same pathways. But the question we're trying to answer, and I'm sure a lot of other people are trying to answer as well, is whether, or should at least, answer is whether they act in, what's the best list of drugs that act in different pathways. And so then you have 2 filters. You have the filter that they have a high effect size, and then you have the filter that they have different pathways that they act in. And then there are other IND, patent enabling filters that you can apply to sort of get to a list of, say, 6 drugs that you want to trial. And then from, at least in our design, which we're going to carry in a few months from now, as soon as the aging clock part of our project is finished, you want to look at combinations of 2 and 3 out of those 6, right? And if you have combinations of 2 out of 6 and combinations of 3 out of 6, all of the possible combinations, all of the possible permutations, you have about 35 treatment groups. And that's huge, right? Like if you think about the classical trial, if you have one drug that you're trying to find the effect of, you usually have 1,000 subjects in the treatment group and 1,000 subjects in the control arm. And if you think about 35,000 cats that you have to trial, that's a huge budget that probably nobody's going to give us. But the really nice thing about this is most of these drugs have passed phase 1 trials, which means they're safe enough to use at specific doses in the animals that they've been trialed on. So we are only filtering for ones that are trialed in at least one species of felines. And so the next question is, okay, well, what do you do about, maybe you don't have to do as much safety trialing on those 35,000 cats, right? Well, how do you lower the sample size? Because that's really, or decrease the budget per cat. And then our answer to that is that statistics is really about finding the adequate sample size to demonstrate an effect of a specific size. So if you have a very modest effect, like 3-5% life extension. If you want to measure something that's so small and that some large error could mess up your understanding of whether that experiment was real or not, was not real or not, well interpreted or not, then you have to have a very high sample size. You have to have thousands and thousands and dozens of thousands of people against thousands of people. Whereas if you're trying to measure a very high effect size, for example, if you're trying to measure whether men are on average taller than women, which is a very high effect size in the population, you don't need the sample size of hundreds of thousands of people to confirm the effect. You can actually confirm that effect just by watching people as you cross the street for like 2 minutes. So you just a couple dozen people, if you look at them, you will observe the effect. You will observe that on average men are taller than women by, I don't know, like 10 centimeters or whatever. And that's the same case for us as well. We want to run 35 treatment groups, but we don't have to have 1,000 cats in each one of them to be sure that the effect is real, because we're not looking for low effect sizes. If there's some low effect size, like 7% between 7% lifespan extension between whatever drug combo, we don't care about it. We don't need to notice that effect. If we see an insignificant p-value on some reduction there, we don't care to look further into it because we know for sure that it's not going to be a high effect size. Whereas for evaluating a high effect size, really from our power calculations, you really only need about 50 cats per treatment group. As long as provided you look for high effect sizes, that's the target of the trial, which it is. And so if you multiply 35 by 50 cats, you get like 1,750 cats, which is a moderate trial of moderate size and really fundable and really something that, for whatever reason, nobody has done so far. So people have done combinations of drugs, but they haven't done true random permutations of the drugs to really test the hypothesis that they act in different pathways and find the best combination. Yeah.
Yes, thank you for that answer. And just to follow up on an earlier comment that you made, Ben Ballweg kindly provided us with a link to this page on the African buffalo. So it is conceivable that some lions in Africa would hunt these particular kinds of buffalo. Now, I'm wondering, Jose, do you have any follow-up questions or comments for Alexandru?
Thank you very much, Alexandru. We are looking forward to seeing the results. So what is the time frame of what you expect to achieve? We are talking about 2 years, 5 years. How long, please?
Way shorter than that. We at least on the initial plans, initial roadmap we have, we want to finish the aging clock study in the next 3-4 months or so. And right after, we want to start the trials, conditional on getting a funding. And that's what I'm struggling with in the past few weeks. I've been trying to reach a lot of people, a lot of investors to sort of get funding for this, that we can start as soon as possible. The nice thing about the study is that it's based on biomarkers. So we're going to try to find combinations of drugs that improve sarcopenia in elderly cats. And we're also going to measure their aging clocks so that we can see, is this also a nice therapy for aging in general? Maybe, or maybe not. And so because we're only using immediately measurable things like aging clocks and sarcopenia CT sort of measurements, and like MRI and whatever other things the vets we're currently discussing it with are suggesting. I think the timescale is that we're going to get recruitment starting in September or October, and the trial gets started somewhere around November. And hopefully we're going to have most, if not all, of the data by January 2023. So we aim to move as fast as possible, mainly because I'm not interested just in cats. So because this thing isn't happening in humans right now, my interest is in doing a proof of concept and growing a company in the pet healthcare side, and then convincing bigger investors to give us all of the funding we need to go in the human arena. Because in the human side, you really need an army of lawyers to start anything. And you need a lot of funding and proofs of concepts to sort of get it started. So we aim to do it as quickly as possible in animals, so we can move it in as quickly as possible in humans. So my best guess right now is that we're going to finish by January 2023, provided that we get funding in the next couple of months. And the funding is really little. Like if you think about Celine Halioa's Loyal has about $50 million in funding to do these sort of not these sort of trials, but longevity trials in dogs. And as far as I know from their team and people that work there, they're looking to develop aging clocks and also like better fine tune them to dogs and to find single molecule drugs that affect that clock, as far as I know. But Rejuvenate Bio also has a few dozen $1 million. The trial we're talking about for January 2023 is really inexpensive. It costs, in our projections, around $300,000. And the main reason why we're going to move that fast is the fact that we don't need to employ people to do the study. We can act through centralized research organizations, CROs, that you can pay to provide reagents and specific experiments that you need for your trial. So you're only the planner of the trial, but you're not carrying it yourself. You're not employing people to do that. And that's one strategy that I've learned from a good friend of mine from Oxford, Sebastian Aguiar, Brunmeier. And the second reason why we can move very fast is because there's a hunger for innovation in the space and because there's less regulation on it. So while most human things, you think about 2030 or something like that for cats, I think at least the initial trials at the first few centers are definitely going to be done this year, provided funding.
And Didier Crenel is interested in a clarification. What kinds of results are you expecting as soon as January 2023? Would these be epigenetic results, for instance?
So we're expecting to have all of the before and after clocks measured on 35 treatment groups for about almost 2,000 cats for combinations of known aging drugs. So right now the drug list isn't decided. Again, if you have advice on how to best design this trial, we're not inflexible people. So email me, email my co-founder, and email any of us and come with advice about what drugs we best choose in the final list. But we're mainly going by bioinformatic approaches and biochemical approaches to sort of filter down the list into what final 6 or 7 drugs we're going to pick. And then what that list has to be is drugs that we highly suspect act in different pathways. And if they do act in different pathways, then any combination of 3 of them should improve lifespan by a lot, provided that the pathways are antagonistic. So by 2023, what we hope is that one of the 35 treatment groups is going to have a very high effect size on the lifespan clock that is significant even after multiple comparisons correction, and that we can sort of recommend for a larger trial to say, okay, let's confirm that this is actually happening. We have this combination of drugs that gives significantly more lifespan extension than just any individual drug. So say you give 3 of them, if you get 40% lifespan extension in cats. That's really great. That's a product that that would definitely hit the market. If you get more than 50%, I'm really afraid to say that because I don't want to spoil it. But if you get anything than that, then it would be a premier in aging overall. And I really hope that would happen, even though I don't want to promise it, because really it's not in my control. It's really biology. And if it does, then I hope to get connected with investors like Laura Deming. I look up to, TheelCap, sort of high level investors that can really fund this, because in the human side, it's really capital intensive. You cannot do much without dozens of millions, if not hundreds of millions of dollars. Whereas in cats, I think it'd be really nice for people to see Sebastian Aguiar's recent talks about longevity biotechnology venture capital. And one of the things he says in those presentations, he works as a venture fellow for Apollo Health and a bunch of other venture capitalist companies. Essentially, he says that startups have a way higher efficiency per dollar spent in terms of drugs approved. So a large pharma will usually get one drug approved for $1 billion, for $3 billion, depending on the way you're modeling the data of expenditures. In startups, you can get, on average, a drug approved for just $80 million. So what happens a lot these days is that a lot of big pharma companies are just buying the best startups, and so this is really the landscape right now. And I think we have an edge as a startup that we can move quickly and we can do stuff with very little funding, and hopefully convince investors in moving into this sort of stuff into humans.
Of course, in the long term, if you do succeed, and I hope you do succeed on the time frame that you discussed, and spectacularly so, one of the big risks of a big pharma acquisition that many of us in the longevity community have become quite cognizant of recently is that big pharma tends to be a lot more conservative, not politically conservative, but cautious in terms of their objectives, and might not want revolutionary change in the healthcare field. So if a startup might be more ambitious and seek a radical life extension for cats, if not for humans, though some do seek radical life extension for humans, big pharma will look at the quarterly bottom line and say, well, how can we maximize our short-term earnings, and not really look at time horizons much longer than 5 years, if we're even lucky enough for a big pharma company to have that kind of time horizon. So how would a startup, in terms of its strategy for growth and revenue, avoid essentially falling into the trap of capitulating to that conservative big pharma mindset?
So I think the main reason why they're able to, I don't know how to go forward, but I can say what things are already in place in terms of the developments, is the fact that most of the people that start startups in the pharmaceutical space started because they care about that approach, or because they know it's very effective, even if they don't care about it. Whereas in the pharma space, you have all of the incentives and lack of the incentives of any corporation, really. And I'm very young. I'm not born into a billionaire family, so I don't know much about corporations really at that level. But from what I can tell from writings, from books on this topic, because I'm very passionate about pharma and venture capital and biotech space, I think the only thing that can sort of keep startups from doing stuff for the short term is their team's motivation and the fact that they care about the product or effects or efficiency of their approach. I might have to leave for just 30 seconds, but I'll be right back.
All right, that is fine. And while Alexandru is away for a moment, I would like to highlight a few comments that were made. Mike Lazine agrees with my statements. He writes, Big Pharma cares more about putting Band-Aids on ailments and similar kinds of approaches than cures for anything. And Jason Mallory writes that Kaiser Hospitals is actively avoiding AI diagnostic tools, and this is an example of a conflict he perceives between the desire for short-term profits and medical progress. But thank you, Alexandru, for your answer to that question, and certainly the motivations of individual startup founders and team members are crucial in avoiding capitulation to that mindset. Of course, do be careful about how you structure your agreements with investors, venture capitalists, and so forth. Try to set up those agreements so that you as the founder cannot be ousted. This is especially a risk if the startup becomes spectacularly successful, and we have seen far too many founders lose their positions precisely because they did originate a breakthrough concept that somebody else wanted to appropriate. Now, I understand, Alexandru, that your time is a bit limited right now. I want to get Art Ramon and Ben Ballweg the opportunity to ask some questions or make some comments. So let's start with Art Ramon.
Yes. Do you anticipate any problems with animal rights groups like PETA, trying to argue that, cats are sentient beings and they're not consenting to becoming, chemically modified organisms? And do you anticipate any problems with these cats getting loose and becoming feral and becoming, just a grave, damaging invasive species to native bird populations? And if PETA doesn't convince anyone to, recognize cats as sentient beings, wouldn't it make sense to sterilize the longevity cats so they don't become a problem as a invasive species?
I think to the last part of your question, I think that's really, it's really hard to figure out how much of that is because really, this is a U.S. Transhumanist Party discussion. Maybe we can bring a bit of politics into this. I think it's really interesting to think about how much of that is a negative externality that has to be mitigated by some centralized authority, or how much of it is something that has to be managed at the local level, or how much of that is just freedoms of individuals that you can't insurmount with just power, governmental power. So I think it's a really hard debate, and it's very political, and it's really a thing where I'd like to get my popcorn and watch a bunch of minarchists and libertarians fight socialists and communists and centrists fighting both of them. And I would love to see that fight. I would probably even pay money to see it. But I wouldn't give an answer because my own opinion fluctuates, as most people's. And also, I don't want to fix it in time and say, oh, someone can point to this and say, oh, Alex is this on the spectrum. And also, I think it's a serious debate. And I can't really participate seriously in it because I'm not a scholar of politics, even though I'm very interested in it. And really, in terms of castrating cats, whether they're humans or not, actually, I think most humans don't care if cats are people or not, whether they have rights or not. They just don't want they just don't want people in general to be, mistreating them physically right? Like beating a cat, something like that's unacceptable to most people, and probably rightfully so. But then in terms of castration, that's definitely something that's highly debated, whether it's voluntary or not. I think it's an interesting way of thinking about driving the dynamics of predators and negative externalities like ecology. And cats are definitely a big threat to birds in general, and to diversity, ecodiversity in general, since birds drive some of the other ecological dynamics in the area. But how that should be managed, I have no idea, really, or at least I don't want to pinpoint it. On the other hand side, talking about PETA specifically, again, I don't want to pinpoint an opinion on PETA, but I just want to make it obvious to everyone that's watching this that obviously, as all organizations, PETA is doing both good and bad things, right? Like you can single out Bernie for saying something about Castro. Castro did something good, right? Or something like that. And it's like, if Castro is telling you, breathing is good, you should breathe, then obviously you don't want to go all like, oh, commies told me breathing is good, maybe I shouldn't breathe, or something like that, right? So like all organizations, PETA is doing some things that are good, right? Like they're fighting some, and not some, like some significant injustices done to animals. But at the same time, as I guess from your tone, you've observed that they do some pretty nasty stuff to people that are actually not against animals' rights. And I sort of agree there. I've seen some violent things happening related to PETA. And I don't know if that's individual actors or the organization itself. And again, I don't want to, like pinpoint myself, oh, Alex agrees with PETA or it doesn't agree with PETA or stuff like that. But essentially, I think they are a problem, especially if you're doing something bad. Like if you're doing something that's not bad to cats, you're probably a problem, or some rogue individuals inside them, or maybe the actual organization, they're going to be a problem or not. But if you do something bad, it's definitely going to be a problem. So there's been news recently about federal funding for whatever experiments where cats were killed at the end of the experiment. The reason why they were killed is they weren't pets that were owned by or that were in terminal diseases or anything like that, but they didn't have owners. They were specifically bred just for research. And that's what happens to a lot of mice, and people don't even know. Most of the mice at the end of experiments, unless they're on a survival curve experiment, they're killed. And especially if they're like a control that outlived whatever treatment, those ones are going to get killed at the end of the day because it's cheaper that way. And people care about the economics of animal trials. Well, the people that ran those sort of studies. For us, I'm sure it's not going to be a problem. So even if there are rogue people or PETA itself that takes an issue with the sort of research that we're going to do, I'm sure it's not going to be a problem, mainly because we're not doing research on cats to make research on humans better. We really want the drug to be approved for cat uses. So we really want to mitigate cat sarcopenia. We want to mitigate chronic kidney disease if we are successful with sarcopenia. We want to go into a few diseases and sort of try to get new therapeutic approaches for them for cats. We're not trying. It's not the classical, I'm trying this in mice so I can do it in humans sort of thing. We really want this to work for cats first. And secondly, and we don't expect that the cocktails are going to work best for cats are going to work best in humans. We're just hoping that the trial design is going to work nicely in humans. And then the second thing is, we're not going to kill cats. I am 100% sure that we're going to manage the drug doses such that they're known to be safe from previous trials. So we're not going to do anything harmful to cats at all. If anything, we might prolong their lifespans and health spans. So we're not we're not pushing aggressively some new experimental drug. We are careful about the dosages of combinations, and we're doing a lot of mitigation in there. And once it starts, and our plans are completely non-harmful and are not for selfish reasons. Well, some selfish reasons, but that's not the main driver of it, right?
So how quickly would the benefits of the drugs wear off if it were to escape and become viral?
We don't know. We have to see that in the data, and we are going to evaluate that. How we are also trying to figure out how we can translate from age clock reversals to actual lifespan increases. We're going to do a lot of statistical analyses and nice modeling of data once we can fund the whole of the goals that we have for the company. So right now we have 10K in funding from Gitcoin for roughly 10K for we are getting it on the 15th of April for the first agent clock for cats. They're fine-tuned for cost and as high accuracy and improved signal from other methods than just blood clocks. And then we need about 300K worth, depending on the model, for the trials. And for all the other evaluations, obviously you just raise more money, or you use the revenue that you get from the licensing of the first things that worked. And so depending on what we were going to try to evaluate, and one of the things that we were going to try to evaluate is how fast it wears off. I think that's how fast it wears off is definitely not going to be a part of the data that we're trying to generate by January 2023. So it's going to be a later time point in terms of that.
Yes. So thank you for that answer. And this is a fascinating discussion. Didier Cornell writes that the very strange thing is that even for studies concerning longevity, in most cases, mice are killed before they die, quote, of old age. And the reason that is sometimes given is to better be able to examine the animals. But really, when we are investigating the impact of particular treatments or regimens on longevity, the best practice, in my view, should be to let the mice live for as long as possible, because one might be surprised, including from the control group as well, as to how long these mice can live. But I don't think anybody would be able to beat Andrei Bartka's longevity record for his genetically engineered dwarf mice that almost lived to 5 years, unless they were to let the mice survive for as long as they would survive after the official completion of the experiment. Now, I want to give Ben Ballweg the opportunity to ask his question as well.
I want to clarify, I guess I thought before today that we were going to be talking about why you were developing a test for cat diagnostics, and it seems like the goal is more to test multi-treatment therapy for cats. So if that's the goal, can you go into why you need to develop your own cat longevity clock? Is it a cost issue versus Horvath's, or tell me more there?
There's multiple things there. So I'm going to start with the last part of the question, because that's a really interesting one. So most of the clocks are actually very expensive. I try not to cite specific codes that were given to us, just because they're personalized to each provider and requester and so on. But most of them are above $100 a clock. So if you have to clock something twice, some cat twice, you get above $200 per cat in terms of just the primary endpoint, right? So, or the secondary endpoint, whichever way you designed the study, which is already more than most therapies. So just the readout of whether your therapy worked is more expensive than your therapy, which is very, it's ill-advised, I think. And obviously it's ill-advised in the context of where you can improve that. Obviously, if you can improve that, that's fine. It's still cheaper than a survival trial. So a survival trial will probably cost in the thousands of dollars, depending on how you design it. And the reason why we're trying to do this cheaper longevity clock is because we want an open source clock that's cheap as possible and accurate as possible, as accurate as possible. And there are very few ones like that. Actually, none of that, except a very recent one that came, I think, a few months back, called TimeSeek. And it's made by, in collaboration with David Sinclair's lab, I think. And the idea there was essentially that you could get the same, or at least almost the same signal just by reading less DNA methylation molecules. So they're doing low depth bisulfite sequencing essentially. And they're using a bunch of advances that have been made at that point with dealing with sparse data. That was mostly because of single cell measurements that were improved back then. And I work with single cell sequencing technology at the institute I'm at right now, that I'm graduating from in a few months at the University of Oxford. And that definitely sparked my imagination as to how low the costs can go. So the cost from the TimeSeek technology can go to about $10 a sample, which is amazing, right? And it keeps a fair proportion of the accuracy of other clocks out there. Now, there's definitely disagreements among statisticians and among people that design aging clocks whether that's appropriate. I'm not sure if Professor Morgan Levine, the work of which I admire, if Professor Stephen Horvath agree with that sort of strategy. I sort of think it's interesting, and I think we ought to pursue it. And I think we also ought to pursue things that are outside of omicsy blood readings to sort of improve the signal on that, such as MRI imaging and so on and so forth. And so that's why we're trying to design this new clock. And also because we're fine-tuning it to cat data, which is, if for people familiar with deep learning, it's similar to like a transfer learning process. And you want to do that to the best of your abilities, which people haven't done in cats yet. And what we're doing is we're trying to improve or change some methods from the TimeSeek technology, and bring in a bit of our own ideas and technologies we had in mind. And also, we're very interested in using it for, as you said, combinatorial drug trials, mainly because even though the primary endpoint of those combinatory drug trials is going to be something very simple and classical, like a clinician's evaluation of the degree of sarcopenia in a cat, which is usually done with imaging of the muscles or BMI combined with some body fat percentage measurement or something like that. We actually also want to see if the drug trial the drug combo that succeeded in sarcopenia, which is obviously an age-related illness, also lowers the overall aging clock that's similar to TimeSeek and similar to older aging clocks like Steve Horvath, the Levine clock, and so on and so forth. And so, we're trying to, I don't know how this translates from Romanian to English. I think hit two bunnies with a stone. It's very primitive, but and very raw. And now definitely PETA's going to go after me. But
so in the Western world, they hit birds, and in Romania, they hit rabbits. Interesting.
Yeah, I think, I don't know. It's maybe I'm making stuff up right now, but yeah. De pur cu o singură piatră? Yeah, probably that. I don't even know if that's maybe, yeah. But essentially, that's the reason why we do the clocks, so that we can improve things that are already used for other purposes, not just combinatorial drug purposes, drug trials, but also for the combinatorial drug trials, so that we can see whether it could perfectly be the case that you have a combination of drugs that works very well against sarcopenia, but doesn't really prolong lifespan. So that's why, as opposed to only having a primary endpoint where we're looking at the muscle mass of the cat, we also try to have a very good measurement of longevity that's not as expensive and long taking as survival studies, but efficient and accurate and affordable.
All right. Well, thank you very much, Alexandru, for your time today and for the insights that you've shared. Now, this is a fascinating conversation, and hopefully we can continue it in the future, especially as your research gets further along. But now our time for our Virtual Enlightenment Salon has come to an end. So thank you, everyone, for watching. Thank you to our panel and our audience. And until next time, every Sunday at 1 p.m. Pacific time, 4 p.m. Eastern time, live long and prosper.
Live